Okay, now that everything's actually working. Got it. And going back here. All right, well, we'll go with this. There's nothing really important at the top. Okay, so for real this time, I'm gonna do something a little bit different. I've learned in the past 4 years that I've really liked combining science with being vulnerable and bringing some humor. So that's what I'm gonna try to do here today. And I can only thank all of you for really being able to let me do that, being in such a supportive community, which also means if there's anything that you don't like, it's kind of your fault anyways. So with that, we can get into it. And also, I mean, I feel like you should have expected this coming because I sent out a video of me getting like fake engaged as a pumpkin guy 2 years ago. And if, for those who don't know what I'm talking about, you can actually go on YouTube and see me getting fake engaged to one of my friends. Um, it was great. But anyways, we're here to talk about science. So how did I go from doing all these random odd jobs to actually working in this lab using bioimaging techniques to better characterize and diagnose cancer and sarcopenia? So really, all of it was Plan B. Back when I was being a personal chef, just figured I needed some extra letters behind my name and applied to grad school on a whim. I actually was trying to get into population health, but saw that you could check up to 3 boxes. I just randomly checked nutritional sciences. I had some interest in it. And a few months later, I went to the recruitment weekend and felt at home immediately. Everyone was so welcoming, casual. I felt like I belonged immediately. So when I got the offer to get into the program, I did. I was very ecstatic and decided to admit or attend here, which was fortunate because then I was also rejected from population health. Um, and my first seminar, my first semester of classes, I was taking minerals with Roger, and Roger was talking about paradigm shifts in research. So all these different technical things or advances in research that really advance the fields of biology, biochemistry, whatever you want to think about. And it started making me think about like a paradigm shift for myself. At the same time, I was working in Adam's lab, and I never had any interest in doing anything within muscle, but all the fancy toys of ultrasound where I could see into my leg or DEXA within there and seeing body composition really intrigued me. Um, and Adam started talking to me about like why we're actually measuring this too. So muscle mass, no matter how you actually characterize it, and I should say muscle wasting, no matter how you characterize it, can lead to lower quality of life, disability, and eventually lower survival. Now, we primarily in the lab focus on sarcopenia, which is the progressive loss of muscle strength and mass with aging, typically seen with aging, and also cachexia, which is the loss of muscle mass, sometimes with fat in the presence of disease. And the way that both of these conditions are typically diagnosed involves something with muscle mass. And so Adam would tell me the problem with muscle mass. So even though these are used to diagnose sarcopenia and cachexia, there's a mismatch that Brett Goodpaster started to illuminate back in 2006. And he actually came here to talk. He was the muscle and diabetes guy. He came out with this now pretty famous chart in our field showing that there's a mismatch between when people start losing muscle function and their strength and when they start losing muscle mass. So people start losing a more precipitous drop in muscle function than they do in mass. So there's something that must be going on that's besides just a muscle mass drop. And we're also seeing in the literature, after this, that muscle mass doesn't always actually predict mortality. And we're also seeing that it has its limitation in the way that it's assessed, which I will get into. So what can be in its place? It's something that we think is muscle quality. Now, very ambiguous term, and it can be defined in many different ways. I've got a list here, and it's not even extensive, but it could be muscle mass corrected by strength, mitochondrial function, inflammation, fluid ratios and tissue compartments in the body, or the infiltration of lipids into muscle tissue. So there's nothing consensus about it. but it seems like this is the way that the field is going. And most importantly, muscle quality can still predict function and also mortality when we're thinking about the health outcomes of muscle quality. And when I was in Adam's lab, I also had a personal connection to this too. So on the right is my grandma, and I think she and I probably both appreciate a bit of humor. On my left is my great aunt, and when I was in lab for the first semester, my grandma was going through her second round of chemotherapy for lung cancer. And so I got to see her go through the whole progression of losing muscle, and I really did see her become weaker, more immobile, and eventually pass. So in 2018, Adam and I formally decided to work together, and then we got to work on trying to rethink ways that sarcopenia was diagnosed. I have two groups up here, left and right, that are kind of the consensus, quote unquote, consensus groups that people go to actually define sarcopenia. So for both, the first step to diagnosing sarcopenia is a muscle function test, typically with hand grip strength. So it's just a machine that you squeeze. If people have low muscle or low hand grip strength, then they go into the second step, which is being confirmed with DEXA scans, which I'll also go into, measuring their appendicular lean mass, so their muscle mass in their arms and legs. So no matter what, there has to be a confirmation with muscle mass. To do this, clinicians will use this machine called DEXA, which we have down the hall, and I'm sure probably most of you have actually seen it who are in the room now. But the can't go too far. The X-ray will be emitted from below, received on top, and depending on the tissue, will block a certain amount of X-rays that are going through. With DEXA, you can see bone, fat, and lean mass, different colors. So white is bone, fat is yellow, lean mass is orange. This is me definitely before the pandemic, but this also has some issues because lean mass, and typically what's seen in the clinic as muscle, shares a lot of the same attenuation properties as water. You can actually just throw a bag of water onto the DEXA machine and DEXA will read that as muscle, which is a problem when you start to get older and your fluids start to be retained in the body. And if you had fluid buildup in your legs or your arms, then DEXA is going to say that's muscle when it's not necessarily muscle. And so when we're looking at appendicular lean mass, so the lean mass in arms and legs, which is used in diagnosis, we need something that can actually prove quality versus just the actual quantity, which might not always be accurate. So we added bioimpedance spectroscopy, or BIS. And so bioimpedance spectroscopy uses a current, it sends it back and forth at different frequencies in your body to assess body composition. At low frequencies, it can't get through cells. At high frequencies, it essentially can. And what that can help parse out is your intracellular here in white versus your extracellular fluid, which is in blue. And we care about intracellular because that's what is associated with contractile and healthy muscle. The extracellular doesn't contribute. And depicted here, you can see the progression from young to old where people start to lose their intracellular fluids. So this can essentially serve as a quality factor to be used with DEXA. And it's a very easy machine. People just lay down. It takes 2 minutes to do. So very easy and convenient and also relatively cheap, especially compared to more fancy techniques like MR and CT scans. So that's all well and great that we can actually have these techniques, but we need to do some sort of real life performance comparison. So in our study, we actually started looking at hand grip strength. Oops. Again, just squeezing this as hard as you possibly can. And then also we were more excited about this jump power mechanography. So people stand on a plate, they jump, we see how much power they can generate. And we were more excited about this because it's a whole body movement versus just a few muscle groups that were in hand grip strength. For our sample, we went to something a little bit bigger than what had been established before. So when Adam was in Minnesota, I believe is when you did this study. So he actually looked at appendicular lean mass from DEXA and corrected it with bioimpedance spectroscopy before. And he saw that compared to the traditional measures of measuring sarcopenia and diagnosing them, this new method of DEXA and BIS actually worked better and predicted hand grip strength and jump power better as well. So we did the same thing, but we wanted to do more people. We jumped from 112 in mostly 70 plus older white people to 248. They're in a more diverse sample. with the same measurements though, so we can compare. We still wanted to validate that the ALM from DEXA corrected by BIS was going to predict muscle function better. And also we came up with another measurement of just using leg lean mass. So just your leg muscle corrected by your leg tissue and see if that could actually predict function even better. We did that in also, if you're curious, we did that in the Midas data set, which is all publicly available data. And there's so much stuff in there. You can definitely go check it out, all for free. So our descriptive stats, our population was mostly women, which is awesome to see. We had not quite an even split between black and white individuals, but definitely closer than the previous study. Age was non-normally distributed, so we made our own categories, trying to parse out some real-life changes in different ages, but they're roughly equivalent between each category. And we had METs, which are metabolic equivalents of tasks, essentially measuring how much physical activity that you can do. A lot of respondents said they do 0, which is impossible. So this is not the greatest measurement of physical activity, but we tried to include it just in case it showed any differences. And we really just made that like a 1 or 0, either they were somewhat active or not. When you were starting to look at bivariate correlations, I found this really interesting. Hand grip strength, which is usually used in the first step of diagnosis of sarcopenia, that is an age-related disease, was not actually associated with age. However, jump power was. And when we start kind of just look at all these different muscle measures, these are the older traditional ones. These are our measurements down here. In the end, our leg lean mass corrected for by the fluid ratio was performing the best. When we actually go into the predictive models, so here we're predicting jump power as an outcome. This is kind of a messy table, but I'll walk you through it real quick. Pretty much all the muscle measures in the model were significant. We have our covariates up here, and then we have our different muscle measures here. I think the most important thing to see with jump power, no matter what muscle measure, they were all increasing. So as you got older, your age was going to significantly decrease your jump power, which should be seen for something that's like characterizing muscle wasting with age. And then when we look at the R-squared value, so this is how much our model is actually predicting this outcome, in the end, our model of leg lean mass corrected with BIS was actually performing the best too, which is pretty neat. And we also did head-to-head comparisons of all these models, and really significantly, our model was out and on top at the end. So to summarize this a little bit, our leg lean mass corrected with a fluid ratio was outperforming even the traditional measures of sarcopenia, so maybe that can be implicated later on. Jump power was associated with age, although hand grip strength was not, and it does actually seem like the ratio of extracellular to intracellular fluids is actually doing something to improve the quality and diagnosis of sarcopenia. So that's really cool, and we kept working on different ways to categorize muscle health. But first, I want to introduce you to another person in my family. This is my mom, and no, the apple does not fall far from the tree. I think there's probably a whole lineage of comedy weirdness on my mom's side of the family. But in January 2020, took an improv class with Amy Zielinski. I highly recommend it to everybody. I wanted to learn how to become a better science communicator. I know improv is not for everybody. There's probably some of you in the audience that would rather fake your own death, which is an option in the syllabus if you want to do it. But one of the activities I found really helpful was trying to explain why we're passionate about our research. So I told the story about my grandma, but then I also added my mom. Because my grandma had passed, I was kind of just thinking of the next person in my family who might be next to pass. And my mom is also a smoker, like my grandma was. And so naturally, I just started thinking like, what could I do to maybe prevent some of the suffering that my mom may experience? It's not guaranteed, but maybe. And maybe you also have family members in the same situation. So fortunately, again, in lab, we had some work that was working on lung cancer. So This time, we're going to switch gears a little bit with our muscle quality measures, but we're trying to see if some other imaging techniques can serve as biomarkers for both sarcopenia, so the muscle wasting with age, and also muscle wasting with disease in cachexia. So when we're looking at the deaths of cancer per year, it's not the most prevalent type of cancer, but it does have the most deaths. It beats out all the other cancers. And for those who are diagnosed with lung cancer, it's estimated that at least 50% will wind up with cachexia, so that muscle wasting, which is a significant amount. And cachexia can occur in all different other types of diseases too, renal failure, heart transplant. So although we are looking at lung cancer here, it does have implications for all different sorts of chronic and also sometimes acute diseases too. Like sarcopenia, cachexia diagnosis is also very messy. So I'm not going to read through all of this table for you. Essentially, I will summarize, some of the most popular ways of diagnosing cachexia. So typically, it's seen as muscle wasting in the presence of illness. However, when you actually go through the criteria, sometimes there's muscle wasting incorporated. Sometimes you may incorporate muscle wasting, or you don't have to at all. And the last one is really just looking at body weight. Typically, that would have been okay. I feel like in the old days, you could really see muscle wasting just on subjective amounts. But as the US population worldwide starts to get a bit larger, then just looking at body weight changes might not actually get to the muscle loss that's happening. So therefore, we need something that can actually measure the muscle health, and especially on people who may not be volatile and able to do something like hand grip strength or jump power for that matter. So we started looking at muscle quality again. Here we're defining it as myosteatosis, which is the infiltration of lipids into muscle. And we think this is a really important biomarker, not only because it's getting more and more attention in the literature, but it also still predicts function. If you have a higher rate of myosteatosis, you have decreased mitochondrial function, and it also increases inflammation. So we wanted to see, can we get to a bedside measure so we can have our nice MRI reference measures, but we wanted to see, can we get something that's a bedside measure like ultrasound that's portable, like we literally take our ultrasound machine on the bus across to the hospital to do our measures. Can we do that as a muscle quality biomarker? So we had a small pilot study of 30 people total. We had 10 healthy young adults with no non-significant medical concerns, ages 18 to 30. We had an older healthy group, so we're trying to compare these two to see if there's any effect from aging. Our older healthy adults with no known significant medical concerns, ranging in 55 to 71 for ages. And then we also had an older group that were undergoing treatment for lung cancer. And then between older healthy adults and older adults with lung cancer, then we could actually see the differences between disease and for muscle quality. So our muscle techniques that we used, our reference measure was proton density fraction, PDFF. It gives you percent fat, and it's from MRI. MRI is non-invasive, no pain, can usually be used on most people. For those who haven't done an MRI, you just lay down on this bed and then you go into the scanner, and after 10 or 15 minutes, then for us at least, we can get a PDFF value or percent fat, and we could do this anywhere in the body, which is also really neat. PDFF was originally originated in liver. Here with one of our collaborators, we can see if you just take a circle, a region of interest, really, you can put this on anywhere and it can give you a percent fat readout. And it does this by actually, similar to nuclear magnetic resonance, essentially MRI is, they just call it magnetic because people were scared of nuclear back in the 70s and 80s. But it can look at the spectral peaks of triglyceride molecules to give you exactly a signal from triglycerides and nothing else, which is also pretty cool. So it's a really great reference measure. And only within the past couple of years has PDFF actually been used in muscle. This is our image that we get. So we are actually taking, for all these different measures, rectus femoris biomarkers. So rectus femoris is a muscle just right on top of your thigh, and we're measuring halfway between your hips and your knees. So something that's really accessible for clinicians going forward too, and also very accessible when we're using ultrasound. So we use two different types of ultrasound. One is echo intensity. So for ultrasound, like you've probably seen in TV shows or movies, we have probes that'll go onto tissues, um, just like this here. And then from those images, we can then further analyze them for brightness. So using B mode ultrasound, we can surround a whole muscle, or in our study, we just took a square region of interest. We will see how gray those pixels are. We'll average them together, and they'll give us an echo intensity. And echo intensity has been around for a while. The higher the echo intensity is, usually the more fat infiltration, scar tissue, essentially the lower quality muscle. And we also had one other ultrasound technique that we're using, shear wave elastography. This is a pretty neat technique that it uses ultrasound waves to actually push tissue and then measure how quickly they bounce back. So it's measuring the elasticity. And we're hypothesizing the higher elasticity, the better muscle quality. Although it's actually somewhat controversial in the literature now, and it's pretty new, only been around 5-10 years, so it's still being established in muscle, but that's why we also wanted to test it. And with this, we can also take our regions of interest, and then it'll tell us how elastic the tissue is. We also were able to get some CT scans for those adults that were undergoing lung cancer. So for committee members, I'm not really going to be talking about CT scan, although that's a lot of my dissertation. I'm going to let Jevin do that because he's talking about that in a couple weeks. We also did biopsies to compare how these imaging techniques can be referenced to histology. So trying to cover our bases with this. And overall, our hypothesis for elasticity, higher elasticity, the better. So this bottom row is essentially the best muscle quality that we can get. We also want low echo intensity and low PDFF, or percent fat. If muscle quality gets worse, lower elasticity, higher echo intensity, higher PDFF. So when we compared between groups, things were a little wishy-washy, which is pretty interesting. So in this table, I have only the things that turned out to have any significant differences between groups. We did look at just the general rectus femoris thickness, and that was different between the groups. And here, this is where older cancer and young healthy, depicted by A and B being different, not being shared, they were different, with young healthy having more, or thicker rectus femoris, which changed for rectus femoris echo intensity, so our muscle quality factor, where older healthy adults and young healthy adults were significantly different, but then older cancer was in the middle. And it changed one more time with rectus femoris PDFF, with just young healthy being significantly different than older healthy. So granted, this is 10 people in each group. Obviously, we'll need to replicate that, but it is something that's showing like, okay, there might be mass differences or cross-sectional differences happening at different rates. So we're going to investigate this a little bit more. We also looked at elasticity, like I mentioned. That was not different between any groups, which was kind of surprising. We looked at subcutaneous adipose tissue thickness, not different. BMI, rectus femoris cross-sectional area, and whole leg cross-sectional area, no difference between any groups. I think this is probably the most exciting finding when we are looking at BMI compared to all of our different muscle quality indicators. It's actually only associated with whole leg cross-sectional area. So that means none of these biomarkers are associated with BMI, which then, if you think about the diagnostic criteria for cachexia, when you're looking at body weight changes, this is just kind of flying in the face of that. But just looking at body weight is not going to pick up. These changes in BMI are not a valid way to assess cachexia. I think this is, of the exciting findings, the most exciting, that we got to see that grayscale analysis with ultrasound, that echo intensity was highly associated with percent fat, based on a reference measure in an MRI at 0.75, which is pretty great in a clinical setting. When we were looking at our histology, there were no differences, which also, and this is, we only did biopsies for older healthy adults, and then older healthy adults or older adults undergoing treatment for cancer. This is not super surprising, as in our field, we're kind of rebuking the idea that histology is going to be the gold standard. There's some estimates, at least for liver biopsies, that it only takes 1/50,000th of a tissue or of an organ. And in fact, some of the estimates to get to an accurate whole organ level, you actually need like 3 centimeters of tissue, which is not going to happen in a clinical setting. So hence, we should use some bioimaging techniques. To summarize this, we have that rectus femoris echo intensity and MR-PDFF are strongly associated. So could echo intensity be an alternative biomarker for myosteatosis? Hopefully, have to replicate and see. None of our typical like mass or body weight measures were actually associated with PDFF. Again, suggesting that the criteria to actually diagnose cachexia needs to have something about muscle in there. And so we're working on another project to see how we can actually improve this association between echo intensity and PDFF, which I'll talk about now. So we're trying to maximize this association. I showed before that it's at an association of 0.75 or 0.5, yeah, 0.75. But we want to get it closer to 1 as much as we possibly can, which if we could, means we could get MR quality images based on something that's really portable. And so that's good for research, but also thinking for facilities across the world that can't afford these multi-million-dollar machines like MRI. Then they could get something that's $15,000 to $20,000 of an ultrasound and start going into really assess a good marker of muscle health. So working with undergrad in my lab, Sujay, who I think is in Zoom, we started to think about ways we could correct echo intensity to get it more like PDFF. The subcutaneous adipose tissue that's on the top of your leg, so still looking at rectus femoris here, we've got a layer of fat and then muscle underneath. That fat can actually distort the image and then also make echo intensity become less accurate. And then also there's inter-rater reliability issues between ultrasound users. So the way that Katie, Jevin, Adam, and I use ultrasound is going to be a little different, which is some criticism to ultrasound. However, that doesn't stop others from trying to correct echo intensity. So there's other group, Yun et al. In 2015, they released a paper with a correction factor trying to assess for like every centimeter of subcutaneous adipose tissue, adding on a certain amount of echo intensity to get it more associated with MRI. We are also in a unique position that in the same pilot study, we had a force measuring device, so we could actually see how much pressure that we're putting on people's tissues. So we're eliminating the compression and ideally getting rid of some of that inter-rater reliability issue. So then all 4 of us can take really similar images for echo intensity. So we were comparing these images between the known forces. So we did 6 total pictures at different forces. So we did 2 at 5 N's, pushing down a little bit, 2 at 10 newtons, 10 N, and 2 at 15. So giving a different measure of compression, known forces. And we actually didn't see between any of the forces in the bottom that there was no differences between echo intensity, which is kind of surprising, but also good for us that we don't have to worry about that. And also no difference in subcutaneous adipose tissue thickness. So compression seems to be pretty minimal on both of these measures. So Young developed a correction factor, still trying to figure out SAT thickness and see how that could affect everything by taking 5 people. And each of these dots represents a different image and a different muscle location. What they did was regression lines for each participant and then averaged all the slopes and intercepts for those regression lines to get an overall correction factor, and then plugged in 1. So essentially in their end, they added 40.53 echo intensity for baseline values to get it to a corrected value, which was actually more associated with MRI in their study. But I wouldn't be talking about it unless I had some problems with it. And 5 people, they didn't know the forces, and they also took measurements at all different muscles, right? So we tried to replicate exactly what they did in our study. They have these really great lines. This was our data, all over the place, but perhaps a bit more realistic, since within a different color representing a different participant, there were known forces in at least 2 images per force. Replicating the same exact thing that they did, we had a correction factor of 78, which if you kind of get the average just looking at this, is around 78, so you're basically doubling. Like that's a massive correction factor. And also takes extra work to get to this point too. Um, so we thought about other ways to try to do correction factors, and in the end, they didn't really do anything. Um, this is our results. I'll jump to the big conclusion here, which is actually good. So we had our own correction factor that I didn't talk about. It's on here. This is when we modeled exactly what Young was doing and then actually used Young's correction factor of the original like 40. They're all around like 0.61, which is the exact same thing of raw echo intensity. For us, we were like, well, our correction factor isn't cool, I guess, but for clinicians, it actually is great. So they don't actually have to do any additional processing. These results will probably change because we're going back and reanalyzing some of the images. We're also probably going to switch our stats around a little bit better since PDFF was non-normally distributed and we're using Pearson correlations which need normal values more than you need to know. But these will probably get better in the end is what I'm trying to say. And now as I'm kind of wrapping up, all of us in the lab have one more study that we're trying to push out and Rachel Fensky will take on. So muscle imaging for clinical application, or MICA. This is like the more and more that we all talk about it, we're getting more excited because it's actually probably going to push the field in some incredible directions. So I mentioned in our pilot study before that we need to replicate. So we're going to do that. We're going to expand our sample to 20 older healthy adults, 20 older adults that are also healthy, but 40 adults that are undergoing treatment for lung cancer. So we're still hoping to get differences between age and due to disease, but within the disease, really start to see when muscle quality might decline and also when our muscle imaging techniques can really pick that up. And if there is a pretty consensus point of decline, that also means a point for intervention, and hopefully which we can also track with muscle imaging. With this, so we're going to have our techniques from last time. So the echo intensity, elasticity, PDFF. We'll have muscle density from CT from 40 people at least this time instead of just 10. But we're also adding all these other imaging and function parameters too. So we will have access to electronic health records so we can predict mortality from these muscle quality measures. Function for both hand grip strength, but also like a leg function test as well. DEXA, BIS, so we can test our same DEXA and BIS correction in this population, compare that to MRI. We'll do histology, and I think also maybe most exciting for this crowd, mitochondrial function, so we can see electron transport chain. So we can do like whole body imaging and also at the same time get to subcellular level, which really, as far as like we know, is the first time that this is happening, and especially combining all these imaging techniques. So pretty fascinating that way. So like I said, we're hoping to really capture these aging and disease comparisons with these biomarkers, when function might change in cancer progression. And I think more exciting recently for me is the crosstalk between bone and muscle. So I did not mention DEXA is kind of the standard for diagnosing osteoporosis as well. And myokines have been shown to affect bone tissue. And so now we can start to capture that at a whole body level. So compare DEXA to MRI to ultrasound and how that affects bone. And then lastly, see how all these biomarkers can predict mortality. So that's pretty cool. We'll keep working on all these different imaging techniques. And perhaps at some point, like this will help those undergoing treatment for lung cancer or heart disease, like my mom. So it's a great story, right? But it's not completely honest, I have to say. When I was in improv class and telling the story about my grandma and my mom, I noticed something didn't feel quite right. And what I kind of came up with as the best answer is because we were asked to talk about our passion in our research, it felt odd to justify my family's suffering a little bit, and also that I don't think I have a singular passion for research. Of course, like it does get me energized. I'm very curious and interested in it, but it doesn't give me the thrill of possibility. What does is all the podcasting that I've been doing. So what started as a way to kind of give back to people during a dark time in the pandemic, by sharing stories really about scientists, about their successes, failures, journeys, scientists of all backgrounds, turned into a few talks, a few uh, people who are working with me on a team, to release consistent episodes, to networking with people across country, to possible workshops with different societies internationally, to a possible career, all of which is pretty absurd. um, I still can't believe it, and it's been in like a year and a half of all that happening. But I think I'm most proud to share moments like this. So Alyssa Adams, that's the one thing that got chopped off. Alyssa Adams and Laura Hernandez have been on. Alyssa just mentioned, like, to be really supported, you have to be able to bring your whole self to your community. Or Laura talking about, for DEI work, it's a marathon and it has to be done over and over and over and consistently. And this doesn't seem like novel information, but it is for science, which isn't necessarily discussed. And I think we've had Joe Handelsman, who's the director of the WID, on there too, and she mentioned, like, even in her job working in the White House, she still felt like an imposter. Not only to her current position did she ever feel like she belonged. So you can imagine, in her 60s, she finally felt like she belonged, which resonates a lot with us graduate students, early career faculty, who always have a little bit of imposter syndrome in the background. So it's nice to bring that on. So I told my mom this, I tell her about my research most of the time, and she can usually follow up unless I get into the nitty gritty of like how MRI works. But I told her kind of jokingly, it's like, yeah, I could help work on your treatment if it ever comes to that. But I think I'd rather talk to strangers. And luckily she laughed a little bit as well. But I did tell her, you know, in the end, because of all this experience, if something ever did come up, I'd still be able to translate what her doctor was saying to her, hold her hand during difficult times, and still make her laugh a little bit. So, with that, I want to thank everybody. For those in lab, you've got a little section in my dissertation for thanks. For everyone in the community, for my thanks, I usually like to write things out. So I did bring a little letter. If I can open it. So, to those in the nutritional sciences community, you have been my home. I immediately felt welcome to the department and felt I would be able to thrive by working with all of you. You've supported me in my education, teaching, business endeavors, and most importantly, in my self-actualization. Some of my earliest and cherished memories I have are making people laugh and supporting them through tough times. By being supported by you, I've been able to take the significance of those memories and turn them into action. Whatever I do now or 50 years from now will involve science, vulnerability, and humor. Thank you for making me feel like I belong throughout. My challenge to all of you is to keep belonging alive. Keep it going for everyone and keep making it better. In the words of Alan Kay, the best way to predict the future is to invent it. So in the end, I did try to do something a little bit different. Maybe there are some things that you liked, some things you didn't like, but what really solidified me in trying to do something like this was an Aristotle quote, which I think is very appropriate to land on for an academic seminar. And it was this quote. So there is only one way to avoid criticism: to do nothing, say nothing, and be nothing. So, thanks. Yeah, any questions? And also we'll take questions about bees if you really need it. Yes. Y- Yeah, yeah, especially in the pilot study, we checked that out for PDFF, and it was pretty surprising. Also tried to look at interactions between sex and PDFF, age and PDFF, and I think because it was at the point with classical statistics that it still wasn't significant, but when just looking at the distributions, it seemed like it was just on the verge. Um, so I think that's TBD really in reality, and hopefully like the follow-up study will be able to do it. But like we have our eyes out for it, and I think that would be really fascinating. And also I was at experimental biology this past weekend, and someone who works with elasticity, ultrasound, was saying, 70 plus is when we start to really see changes in muscle function and elasticity, at least in his studies, which is challenging for us because lung cancer starts to happen more in the 60s. So if we have a comparative age group also in their 60s, it may not actually be the best comparison. Um, so it's just something I think we'll have to keep in mind going forward. Yes, Matthew.
I just wanted to say the first thing is wonderful presentation. I have
Thanks.
one comment, then I got a question. So I wanted to say I really give you kudos for doing your defense seminar the way you want it. Because
Thanks.
when I did my PhD defense, I cosplayed for that while doing it on Zoom and in person. So I give you
Nice.
props for doing it the way you want it. This is
Yeah.
your show, and do it the way you want it and go out because you earned it.
Thank you.
So my question was is with the myosteatosis, that was very interesting. I was just curious, is it known on how it actually decreases mitochondrial function?
The stuff that I've seen, and again, this is,
Can you repeat the question?
oh, yes. Thank you. So Matthew asked, how does myosteatosis actually affect mitochondrial function? I'm going to give a little bit of a cop-out answer in that this is not generally my area, but what I've seen is that higher lipid infiltration within mitochondria tends to mess up fatty acid oxidation. And I'm sure that's not the only thing that happens. Um, but that's at least one point of data they can use going forward.
Oh, okay, no, that's interesting. I didn't know that's a new term for me. I just had one quick follow up. I was just
Yeah.
curious. Is there any data or any research with regards to myosteatosis being related to any of the muscle fiber types?
We haven't looked at that yet, so we're hoping with our follow-up study to, I think we discussed like fiber typing. Um, so yeah, we'll have to see. I think that's the really cool thing about MICA, our follow-up studies, because we're able to capture like mitochondrial function, some histology, and all these functional techniques. Like we can get to that answer. Um, I think where others have done like little bits and pieces to add to the story, we can kind of capture everything at once.
Fantastic. Thank you.
Yeah.
First one for your follow-up study, did you ever consider bringing in a group of younger cancer patients as well? Because I know with different types of cancer, like chemotherapy might be harsher when someone's younger, whereas if someone are older, they try to go a little easier on them, and they still might have that muscle loss.
Yeah. So question was, did we ever consider bringing in a young cancer group? And we have. We don't have a complete like 2 by 2 table of yes and no with cancer and yes and no with age. And I think that's just really a reflection of when lung cancer starts to occur. Um, we have gone with lung cancer because unfortunately it is like a decent model of cachexia, but I think that's it's still on the table and unknown. I think it would be very difficult to get enough people to have a valid comparison without having to wait for many, many years. Not to say it shouldn't be done, but I think it would be really hard to capture. And maybe there's also just a different cancer type that could happen. Um, so yeah, more TBD on that, I suppose.
And then also, I noticed it looked like you had food diaries in the study, or was it not?
Um,
There was a diary in there, but I wasn't sure what it was.
I don't remember.
Okay.
Um, do you remember in the context?
Yeah, so it was you had like a chart that showed all the different metrics that you were
Ah,
collecting for
yeah, yeah.
everyone. And I
Okay.
was just wondering if you had looked at food diaries at all with the muscle listing to see
Mm.
like was there caloric intake reduced or like how you would improve that
Yeah.
or no?
So Deandra asked, did we look from the minus data for DEXA and BIS? Did we look at food diaries since it was actually available? Um, Midas had somewhat of a like healthy eating index that we didn't feel was the best. We did actually look into it and we're hoping to see within Midas, since we have 248 people, could we compare healthy eating index and also maybe explore diabetes? Diabetes also contributes to muscle loss. And I just don't think we had the numbers to really show anything. We tried to manipulate that data, like stratifying by different types of diets, healthy eating index, and it just, I don't think was rigorous enough to get to those differences in the end. Which, Dave, for your question yesterday, is nutritional epidemiology helpful or hurtful? You know, there you go. There's just kind of tool, meh, in the end.
I appreciate your family for
Yeah.
family history and personal history.
Okay. Well, yeah. Thanks for sharing, Denise.
Thank you. It was a great seminar, and I think it's a very important avenue looking at muscle quality. Sure. So this is sort of just a looking ahead question. So I think about your research as you're refining assessment tools to able to assess muscle quality. So let's say you're successful because you have them, and let's just get together here, Adam's research. So what kind of treatments are available? What are you going to
Yeah.
do with this information? Because the next step is treatment, and you think we can treat it.
Yeah, I think that's a really good point. Cachexia is difficult to treat because it's not traditionally responsive to nutritional therapies. Perhaps with our bioimaging techniques, we can see something is happening before there's a point to when there's really drastic muscle loss, and perhaps that can be an intervention point. I've spent so much time on the more diagnosis side. I haven't really looked into the specific treatments that could be helpful. In the reviews, you know, when I'm looking at all the cancer or cachexia literature, I think there's a lot of hope for, you know, biochemical solutions, but there's nothing promising at this point, or they're in clinical trials. There's not a solution, I think, for a pill yet. And I think that also advocates a little bit for our techniques too. You know, there's not a one-stop either biochemical biomarker or treatment that way too. So maybe our imaging techniques can be used a little bit before any true precipitous muscle loss would be occurring. Oh, yeah. Yeah. Just based on the exercise, yes. Yeah. Yeah. And some of the like articles that I was reading, like exercise does seem to help. And also, I think it's somewhat controversial at this point, but for cancer cachexia treatment, there's an idea of fasting as well that somehow switches like the metabolism into starvation mode, which helps prevent muscle loss. But there's debate on that, whether that's actually helpful or not.
There's still a lot of work, and I did some of it way back with insulin on growth factor 1 and growth hormone, especially with IGF when you're in, you know, it causes cancer. So,
Yeah.
but they're clearly hormonal stimulants for muscle.
Yeah, we'll see. Okay, we'll go to Zoom. Then you, Tom.
Oh, okay. You have a question for
Alfonso, what's up?
Hey, Ben, I got a chest cold, buddy. I'm sorry I'm not over there with you. I got to tell you, I'm really sorry.
Okay.
So I might cough a little bit. I'm going to go ahead and drop this in the chat as well. But man, heck of a job. Really
Thanks.
happy. So, you know, I've got two questions. One, how is contemporary archaeology related to contemporary studies of muscle mass, sarco, cachexia?
So typical archaeological studies related to typical sarcopenia and cachexia.
Right, Ben, come on. You're a PhD. I hope
Okay.
everybody's laughing over there, but this
Yeah.
is a serious question because Ben
Yeah.
knows that I'm a serious guy.
Right. Yep. Definitely, Alfonso. Definitely. Can I also give you a really academic answer and say that's not necessarily my field? And I can give you an educated guess, but you'd have to really go to the literature.
You know that educated guesses are what it's all about.
That's right. That's right. You know, it would, depending on what your definition of contemporary is for archaeology, I feel like if you'd even go back 100 years, we may not be discussing sarcopenia and cachexia very much because they seem to be diseases of older individuals. So I think by that logic, I don't know if you could really have much of an accurate comparison, though it would be interesting. I don't know if you could get any of like the bone content, but maybe there was also like muscle loss happening in 30s with poor nutrition, poor environmental factors back then versus now. That's my educated guess, Alfonso. Hopefully that's satisfactory.
Here you go, my friend. Let me give you a clue in my view. Do you know the term LIDAR?
I know, I don't remember what it actually means though.
Okay, it's a imaging. It's a way of imaging the planet. So ground penetrating radar is another way of talking about this.
Okay.
And so now the analogy is obvious, right? No,
Yeah.
no longer do we have to do muscle mass studies using those invasive techniques you described. Likewise, for archaeologists, we don't have to go digging around and disturbing the context that makes the artifacts so meaningful.
Yeah, and I think they did that with a recent pharaoh, right? To like recreate the larynx.
Yep.
I think they just kept like the pharaoh in their sarcophagus.
Yep. There you go, my friend. So here's the second question. So this is why you doctoral students out there in nutritional sciences, this is why not to invite a sociologist, a planning professor to your committee. Anyways, so here's the other question. You know, and of course, I asked the first one because I thought it'd make a great podcast episode, you know.
Okay, thanks.
But the second one might also. So, you know, I think that as my colleagues might imagine, we work with faculty. When we were graduate students, there were different reasons why we ended up with the advisors we ended up with. And so your description of how you got into Adam's lab, I appreciate it. Then once there, we often discover things about each other. And so my question is, if you discovered that whether or not Adam had some particular personal reasons for his work, and if whether he did or didn't, you know, if that had some influence on how you moved around.
I'll say yes. Maybe not my spot to talk about like Adam's personal connections, but I think so. Like I mean, when I reflect 4 years of being in here, um, being able to be vulnerable, I think, with everyone that I've come across has only shown me that they're willing to share more information. You get to see them as a whole person. And I think that's what happened in our lab. And I think that's also why our lab works so well. In the most recent podcast episode of Deeper in Data, Adam and I go into this a bit more. But we really do try to, I think, answer the question of like why our working relationship works so well. I don't know if we came to any good results, but I think just having a mutual respect for everyone, being able to share the vulnerabilities like of what I might not know in the research, but also just kind of who I am as a person and vice versa, probably made the working relationship the best that it could be.
There you go, my friend. I appreciate that. And I'm deeply grateful for the many contributions you've made over there on the other side of Bascom Hill. Okay.
Thanks.
I look forward to seeing you down the road.
Me too. Tom.
Yeah, I just had one quick question about your first project.
Yes.
So you were showing R-squared values, and you were talking about predictive models,
Mm-hmm.
which often means, you know, throwing lots and lots of variables in a model to kind of predict an outcome. So I was just curious whether the R-squared values represented associations for individual variables, or if you were testing multiple variables at a time to try to improve your prediction model.
Yeah, so we didn't take like this typical, I think, model building approach. We still try to be hypothesis driven. Though I should also say, Tom is asking a question about our model building process for our first project. Because we had a covariance model only, I think just to cover our bases to see like the initial R-squared value, which if I can remember correctly, was still around like 0.6 or something like that. So a decent amount. just, age, sex, BMI, race. But then we try to stay hypothesis driven for bringing in appendicular lean mass, testing that by itself, leg lean mass, since that was something new, covering our bases that way. And then the appendicular lean mass model corrected by height squared or BMI or typical in sarcopenia. And then we had our correction factor. So I think all of that was driven by hypotheses, but that is something that we could do and go back and add per covariate, which would be interesting to see.
Sure.
So,
I just think it's worth pointing out that the R squared will always increase.
oh, okay. I see what you mean.
So you just might consider other adjusted R squared.
Yeah. Yeah. And, when we were talking to a statistician about this too, like he suggested doing that as well. But I think because we went more hypothesis driven, we kept it that way. And we did, the R squared value comparisons at the end, but we could also do that same thing when we're adding covariates at a time. It would be interesting to see like what's the most parsimonious model, because then that could have applications to the clinic too. And even though like these techniques have been shown to increase like the predictive models of muscle function, it still doesn't mean it's necessarily going to be put into the clinic at that point. Okay. Well, thanks everyone.